Can you hear me? Okay, hello. My name is Diana Wall, and I'm the director of the School of Global Environmental Sustainability at Colorado State University and the chairperson of this National Academy's Study Committee. In my role as chair of this study and as the moderator of the committee meeting, I'd like to welcome everyone here in the room who's in Washington, D.C., and then also everyone who is online watching the live stream or may watching the recording later. The task being undertaken by this committee of the National Academy of Sciences, Engineering, and Medicine is to explore linkages between soil health and human health. The committee's full statement of tasks can be found on the study website, which is available at the link under project to the right of the embedded video if you're watching online. I would like to give some sense of format of this meeting today. As we did at last week's meeting, I will shortly turn to the committee members and ask each of them to unmute and introduce themselves. You can also find a short biography of each member of the study on the study website. I will then turn things over to Carol Laney, who is the program director with the National Academies, to provide a brief overview of the study process. Following her presentation, I will introduce our speakers for today. We will adjourn the meeting at 3:30 p.m. Eastern. In addition, I want to note that this is an open, on the record. Interested individuals and members of the press have been invited to join as observers. This is not a news conference, and we will not be taking questions from the audience. Reporters who would like to talk with members of the committee are kindly asked to touch base with a staff officer, Carol Laney. Also, anyone who would like to submit a written comment or document to the committee is welcome to do so by clicking the Provide Feedback link on the right side of the study website. You can also subscribe for updates about the study by signing up at the National Academy's Board on Agriculture and Natural Resources listserv, also on the right of the webpage. I would also like to remind everyone that this is an information gathering session. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone to be extremely mindful of the fact that the committee has made no conclusions and that it would be a mistake for anyone to leave a meeting today thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report. Moreover, once the staff report is written, it must go through a rigorous review by experts who are anonymous to the committee. And the committee then must respond to this review with appropriate revisions that adequately satisfy the Academy's Report Review Committee and the NAS president before it is considered an official Academy's report. At this time, I would like to ask each committee member, when I call your name, to turn on your microphone and briefly share your area of expertise. Katrina would be the first.
Hello, my name is Katrina Buobara. I'm an associate professor of epidemiology and a physician at the University of California, Berkeley and UCSF, and I have research experience studying the environmental influences on allergic disease. Thank you.
Joseph.
Joseph Aweka, Texas A&M University, specializes in food science and link between food science, nutrition, and crop quality.
Sam.
Hi, my name is Sam Banerjee. I'm at the North Dakota State University, and my lab, we explore microbial contributions to soil health, but we also look at micro interactions and basically how microbial communities contribute to agricultural sustainability.
Thank you. Sarah.
Hi, Sarah Collier at the University of Washington in Seattle. I am a food system scientist focused on bringing together natural and social sciences and looking at the impacts of cropping systems on both environmental and societal outcomes.
Carlotta?
Hello, my name is Maria Carlota Dao. I am an assistant professor at the University of New Hampshire, and I study the interplay between nutrition, the human microbiome, and human health with a focus on chronic diseases such as type 2 diabetes and obesity.
Mike,
This is Mike Grusak with the U.S. Department of Agriculture Research Center in Fargo, North Dakota. I'm a plant physiologist by training, but have also worked extensively in human nutrition, looking at nutritional quality of foods and the use of foods to provide nutrients and non-nutrients for humans.
Hi, I'm Callie Neal. I'm a professor in the Department of Animal and Food Sciences, where I work in microbial food safety and conduct research in the area of pre-harvest food safety, looking at transmission of viruses, bacteria, and protozoa, and their survival in soils and transmission onto crops.
Uvla.
Hi, I'm Ylva Leckberg. I am with the University of Montana and MPG Ranch in Missoula, Montana. I'm a soil ecologist by training, focusing mostly on mycorrhizal ecology and agricultural management practices.
Thank you, Rebecca.
Rebecca Nelson at Cornell University in the School of Integrative Plant Science and Department of Global Development, and contributing to the food system and health part of the public health school. I have mostly worked on crop genetics, but with an emphasis on mycotoxin aspects, and then more recently shifting to the circular bionutrient economy. Thank you.
Kate on Zoom.
I'm Kate Scow, professor recently retired of soil microbial ecology at University of California, Davis. And a lot of my research is on the microbial underpinnings of soil health and also of bioremediation, biodegradation, as well as impacts of management and agriculture on soil biodiversity and health.
Thank you, Kate. And Schoolis Ray.
Hello, I'm a assistant professor at the University of Arizona in the School of Nutritional Sciences and Wellness. I direct a research program to study nutrition and dietary supplement interventions to improve risk markers for cardiovascular disease and improve inflammatory responses. And I'm moving more into food production systems lately.
Thank you, Anne. Lindsay.
Hi, my name is Lindsay Slaughter. I'm an associate professor of soil microbial ecology and biochemistry in the Department of Plant and Soil Science at Texas Tech University. My work focuses on soil health and plant-soil interactions in semi-arid agricultural systems.
Thank you, Kelly.
Hi, everyone. I'm an associate professor at Colorado State University in the Soil and Crop Sciences Department. My lab works both in soil and gut microbiomes and looks at microbial metabolism and how nutrients flow through ecosystems.
Thank you, Kelly. Unable to join us this afternoon is Nick Basta. And as I said earlier, you can find all these bios, including his, on the website. I'd now like to turn over things to Carol Laney, a senior program officer with the Board on Agriculture and Natural Resources at the National Academies and the study director for this study. Carol will give you a brief overview of the National Academy's study process.
Thanks, Diana. I just want to briefly go over how the study process works here at the National Academies for people who might be joining us today who aren't familiar with it. So next slide, please, Kat. The National Academy of Sciences was established by a bill passed by Congress and signed into law by President Lincoln in 1863. Later, the Academy was granted the authority under an executive order to establish the National Academy of Engineering in 1964, and then the National Academy of Medicine, formerly the Institute of Medicine, in 1970. Today, we are the National Academies of Sciences, Engineering, and Medicine, and we fulfill two roles. One, there are elected members of each of these bodies, joining those societies by an election from their peers for their accomplishments in science, engineering, and medicine. But we also work with scientists across the country to come together for projects such as this one to provide advice to the nation. So our mission is to investigate, examine, experiment, and report on any subject of science or art whenever called upon by the government. And as I mentioned, we fulfill this mission through several convening activities, including consensus studies, such as the one that we're here for today. So I'll just share a little bit more about that particular activity. As opposed to other forums that we organize, such as workshops and roundtables, consensus studies are conducted by a committee that's specifically convened for a unique statement of task. The experts who serve on these committees do so as individuals, not as representatives of any organization or institution they belong to, and they serve as volunteers. Consensus study members review the state of scientific literature to write a report that responds to the problem identified in the statement of task. And then that report will hopefully be used to shape sound policies, inform public opinion, and advance the pursuit of scientific pursuit in the field of question. A consensus study goes through many different stages. So if Kat, you would go to the next one. The first one is when the statement of task is defined between the national academies and the sponsor. The study sponsor in this particular case is USDA's National Institute of Food and Agriculture. Next, we solicit nominations for experts that can complete the statement of task. And we seek to retain a balance of perspectives as well as expertise when we put together these committees. Once a committee has been assembled, it is elevated to the president of the National Academy of Sciences for her approval. And then a committee is provisionally appointed until it has conducted a discussion of conflict of interest, disclosure, and balance at one of its early meetings. After being appointed, the committee moves into its information gathering phase, and we are assembled here today for that process. We invite in speakers to present in person or via Zoom, and members of the public may also submit information to the committee. As it gathers information, the committee will begin deliberating on how to best address its statement of task. Later, they will begin drafting a report. When they have a draft report ready that they think responds to the statement of task, it is submitted to the National Academy's Report Review Committee, and a set of experts similar to those here assembled today will be recruited to provide anonymous review comments on the draft. The committee must then assess those comments and respond to the report, either by revising it or considering those comments. And then once the revised report is complete, it is submitted back to the report review committee within the National Academies to decide whether the committee has adequately responded to the review. When the report review committee says that it has, the report is made publicly available and delivered to the sponsors. I just want to say a little bit about our committee meeting process. If you go to the next slide, Kat. When the committee hears from speakers like we're doing so today, we call this an open session. Anyone can be anyone can be present, whether in the room or online. And these meetings are recorded and posted to our website. When the committee deliberates, we meet in closed session, and that in those circumstances, only the committee and staff are present. This particular study, we are just getting underway with our information gathering process. Next slide. And so we are at the early part of that second bar. As we go into the fall, we'll be working on the draft, as I said, and submitting it to review. And our anticipated timeline is to be done with the report in the spring of 2024. Just give you a moment to look at the statement of task, but you can also find this on the website. Next slide, Kat. And as I mentioned, USDA's National Institute of Food and Agriculture is the study sponsor, and this study is the result of a congressional request through last year's appropriations legislation. Finally, as Diana mentioned, you can find all this information on our website. So on the left here, you can see where tabs to get to the description of the study, which will include the full statement of task, upcoming events. So we have additional meetings planned for May, June, and July. And when the agendas are developed for those meetings, they'll be published under upcoming events. Also, if you scroll down to past events, you can find our meeting from last week, and this meeting will also be recorded and posted shortly. And also, if you need to get a hold of the study staff, my address as well as Catherine Kane's are here on the website. So you can reach out to us there. And I've also circled where you can click to provide feedback to the committee, and where you can subscribe for updates to hear about upcoming meetings. So with that, I'll hand it back to you, Diana.
Thank you very much, Kara. Now it's my pleasure to introduce our first speaker today. Dr. Megan O'Rourke is a Brookings Congressional Fellow with the Office of Congresswoman Shelley Pingree, an agroecologist with scientific expertise in conservation, pest management, and climate change. She serves in the Institute of Bioenergy, Climate, and the Environment in the USDA's National Institute of Food and Agriculture. She holds a PhD in ecology from Cornell University. Her longer bio can be found on today's agenda. Thank you so much for joining us today, Megan, and to share some information about this congressionally requested study.
Thank you so much for having me. Is this on?
I think so.
Thank you so much for having me here. But the purpose of me coming here today was to really provide the background. So where did this money come from? Where did the study come from? What's that history of how it's evolved over time? And what is my Representative Pingree, as well as stakeholders, really hoping to get out of this report? Next slide, please. So the origins of the idea of this report really started back in 2018. And so the Soil Health Institute convened a conference. And the purpose of the conference was really to start exploring this idea of the links between soil health and human health. The main results of this report were really that we need a very transdisciplinary approach. We need to get out of the silos. We have health experts in one area, agricultural experts in another area, and how do we get people to really come together and to talk about those intersections, and not just in a natural science sense, but the social science, the people and the environment together. And one of the main outcomes of this, as we all know, like, we need more research, right? One of the next steps from this conference was to develop a critical review of the literature for publication. Next slide. So Representative Pingree took up this call for study. She has been working on a Agricultural Resilience Act, and this is really a comprehensive bill to address climate change in agriculture. She has introduced this bill first in February 2020, and so has been working on this for a number of years. As part of that comprehensive bill, this is like a 200-page bill, is the request for this NASEM study. And so it includes research ideas, soil health, farm preservation and viability, pasture-based livestock and manure management, on-farm renewable energy, and food loss and waste. So these are just different titles in this large bill. And over 42 different stakeholder groups have supported this, and they're both from the agriculture and environment organizations. Next slide. Okay, so I'm learning this all in Congress. That's my point of being on the Hill, is how do ideas become laws? How do things happen? How does the money flow? So when a bill is submitted, it may or may not be passed in its entirety. In some cases, bills are passed to be incorporated or submitted to be incorporated into larger bills, like the farm bill that's being passed right now. Pieces of bills can also be put into the appropriations process. So laws authorize things to be done, and appropriations funds stuff to get done. So a piece of this Agricultural Resilience Act was pulled out of the big bill and put into appropriations language. And so the money that was appropriated came, and it says the committee's to provides $1 million for the secretary to enter into an agreement with the National Academies of Sciences, Engineering, and Medicine. So that's where the money for this report came from. And the articulated topics within the appropriations include the following: links between human health and soil health, connections between human microbiome and soil microbiome, interactions of humans with soils, identifying linkages between soil management and the nutrient density of foods for humans, and how to best leverage healthy soil management to maximize benefits and minimize adverse impacts on human health. Next slide. Okay, and so then there's all the stuff that's happening in the world between the start of this back in 2018 and where we find ourselves now in 2023. One of the big things that's going on in Maine and in Representative Pingree's district is PFAS. And so this is a class of fluorinated man-made compounds, thousands of them actually, and they are in all sorts of materials. So they're resistant to water and grease and heat. So they're in food packaging and clothing and cosmetics and toilet paper and all sorts of stuff. And they have lots of adverse health effects. So cancer is high cholesterol, thyroid disease, liver damage, asthma, allergies, decreased fertility, newborn deaths, low birth weight, birth defects, and others have been found. And so the link with agriculture is that since the 1970s, biosolids have been applied for fertility on farm fields. And so we're finding now that this has led to contamination of both the soils and in plants. It's been detected in milk. And so Representative Pingree was able to appropriate $5 million to USDA to begin researching this problem, as well as $8 million to EPA for research in terms of drinking water standards and also standards out there for agriculture. There's been all sorts of headlines, right? We have Maine farmers heartbroken after EPA admits that almost no amounts of PFAS is safe. More than 50 Maine farmers impacted by PFAS. PFAS devastates a Maine farm, but poses a much wider problem. And so this is just an issue that's of serious concern that's really causing significant economic losses for farmers in Maine, and is playing into the ideas of what the representative would like to see in this report. Next slide. And so there's a website, and NIFA has outlined areas of work. And the representative also has her perspectives of what she would like to see in this study. And so as this committee has come together, the representative reflected on what she would like to see, again, keeping in mind that this has been an evolving process over the last 5 years. And so going back to stakeholders, we readdressed what we would like to see in the scope of this study. And so what I've outlined here in the green boxes is the pretty straightforward things that the representative would like to see: soil health and contaminants with the PFAS, and also soil health and nutrient density, which is outlined in the appropriations language. And then we have kind of broader areas of interest. And so we have not just soil health, but we have production systems, trying to understand how production management leads to soil health. And I know there were some previous questions about, we want to keep this to agriculture, soils, like what is the scope of system? But the original intent really was agricultural systems. And then we have impacts on communities and people. And that is broad, right? There's a fair number of indicators, like what is the target of human health that we're looking at? And so, you know, ideas about farm workers and rural communities and consumers and food access. And I didn't draw a line to them because you can imagine all sorts of direct and indirect effects between all of these different topic areas. Next slide. So where's the takeaway in this? Again, with the letter, we're very cognizant of the fact that breadth and depth of topical areas are kind of at odds with each other, that a practical report needs to be made. And so how do you go from these kind of big ideas to something practical? And so we'd like the committee to keep in mind that this has been a many-year process. It's not like here's exactly what we want to do and this is stood in stone forever, and clearly this is exactly what we want to have happen. We're not trying to micromanage the committee in that way. What we do really want to see is a transdisciplinary approach. We don't want, like, say, for example, just a natural science approach to the question between soil health and human health. We want to be more broad, reflected in that previous slide with the kind of ideas of what it means to have health. Is it communities? Is it mental health, physical health, that whole package. So trying to utilize expertise from a wide array of disciplines, sociology, economics, public health, as well as kind of, I think, traditionally what we've been thinking about as soil science and cropping systems and health sciences. The representative, as I said, has core areas of interest. This PFAS issue is really an important topic right now. She's been very interested in like systems and nutrient densities and the nutritional value. So taking that systems level to make sure that integrating larger dynamics than say specific group of soil microbiome and gut microbiome, for example. And again, we realized that this is a big task. So for example, maybe it's not a full literature review of all these topics per se, it's a case studies approach. It's illustrating what these novel connections are, because part of what we want to see is a foundation for a research priorities moving forward. So how do we push the discipline in a way where we're intersecting and having a transdisciplinary approach to these kind of big questions that, if we think, what is the average person interested in? Not just like, what do I as an academic find really intriguing in my space, but when somebody thinks about agriculture and human health, what is going to be of interest? What is going to be surprising? What's going to push the field? So with that, I appreciate being able to share the representatives' perspectives here.
Thank you so much. We really appreciate this. I'm going to open the committee for questions. If they have questions, now's the time. We may have 10 to 15 minutes or so that we can discuss this with Megan. And again, I thank you for coming. Please remember to turn your speakers, little red buttons on, please. Okay, I can ask a question. You say this has been a multi-year process. And then we also have this thing, which I think we are considering the transdisciplinary approach to be broader. Maybe I haven't gotten to economics yet, but that's because I may not understand the economics and where you're going with that. Is that what the production systems, whether or not the soil is healthy to give production systems that would be more economically based in that community? Or can you give me a circle on this? This may be this circular economy type thing, but if you could help me with that, I'd appreciate it.
Thank you. I think it might help to flip up to the previous slide, if we can. So I think it's important that we don't want to be really prescriptive. We have a big group of folks in this room who have expertise and perspectives. And so I don't want to dictate a specific answer to that. I think it's more like, here are topics. We hope that the committee is considering the interconnections between all of these. I think we can imagine how economics would play into things such as production systems, soil health, food access, and what that means for the health of consumers. But again, we're not here to write a table of contents for you.
Thank you. Your chance.
I'll jump.
Oh, Mike.
Go ahead, Ann.
I was just going to ask the question of, because of the technologies in the food production systems and the tone of what you were suggesting with novel connections and pushing the field, is it of interest to also explore alternative methods such as hydroponic and aquaponic systems as a way to sort of alleviate the stress on soil-based food production? I know we're interested in the implications of what's in food and not in food as a result of these different systems.
Again, I'm not going to I may not be the most helpful answer because I'm not going to say yay or nay to any specific thing. I do think that the context of what was originally imagined was, agricultural systems in the field in terms of soil health. But I think if there are novel areas that make sense for the committee to address, then again, it's open for the consensus of the committee to decide.
Thank you.
Mike?
Well, let me follow up on that. We appreciate you that I think we all appreciate the fact that you don't want to write the table of contents for us. Your model that you had up there talked about production systems and, hydroponics, aeroponics, those things, vertical farming are sort of gaining. I know you don't want to say what we should, but can you speak a little bit to the breadth of production systems that were envisioned as this went forward?
Yeah, I think there was a question at the last meeting of what system scope would entail. So there was talk about, say, natural systems and soil health there. I don't think that was in the original scope. We did talk about animals. We did talk about plant and cropping systems. We didn't necessarily specifically talk about the alternatives of the vertical farming and the hydroponics. But I mean, again, I think if in the context of those contrasting systems that makes sense to bring up some ideas that perhaps aren't regularly considered, then that can make sense.
Thanks. Other questions, comments?
Hi. The original statement of task, a number of the bullet points talk about the microbiome specifically. And I was wondering if you could provide a little more context on how you envision today the focus on the microbiome fitting into your nice conceptual diagram.
Yeah, I think there's more verbiage about soil health as a whole than about microbiome. Though if we turn back to the 4 bullets of the original verbiage, I believe microbiome is in there. So I think, microbiome is part of the larger soil health system. And so it's important, but it's not the only thing.
I just follow up if I followed correctly on the slides, the focus on the microbiome came in at the point when something got picked out into the appropriations language or something like that, like the original letter or as it was envisioned didn't highlight the microbiome, but then it showed up there as a particular highlight. Do you have insight into that process and the conversation that went on to bring that in as a highlight or a focus area?
Yeah, if you refer to the original Soil Health Institute conference report, it's in there. And so again, I think it's an important part of the whole question of soil health. So how does that fit within the scope of, there's a scope of what you can reasonably do. And so obviously it's not the only part of this that's important, but there's also a finite amount of work that can be done. And so I think certainly the concept of soil health is not restricted to the microbiome.
I wonder if you could, the appropriation was there to put this report together. Could you speak to a bit about what you or the representative would like to see this report ultimately do?
Yeah, one of the specific call outs in the appropriations language is to identify future areas of research. And in the larger context of what the representative is interested in, the Agricultural Resilience Act is a major thing that she works on, and that's all about climate change. And so I think when we talk about soil health, that's something that the representative thinks about is the carbon and the nutrient cycling and such. And so I guess going back to the question of microbiome and soil health, the context she's also interested in is the climate change context. And then I think ultimately, how do we use this report? Who's the audience going to be? How does this spur opportunities in the future? So I think it's some sort of balance between, okay, here's a synthesis of what we know, but really here's a synthesis of what we don't know and where we need to move forward and to bring together currently disparate areas of work into a synergistic whole.
That's very helpful. Thank you. Any more questions? Thank you very much. I'd now like to introduce Dr. Chuck Rice. Chuck is from Kansas State University. He's a university distinguished professor of soil microbiology, and he conducts long-term research on soil organic dynamics, nitrogen transformations, and microbial ecology. Recently, his research has focused on soils and climate change, including carbon and nitrogen emissions. And that's been in both agriculture and grasslands and soil carbon sequestration, I should have said, and potential benefits to ecosystems. And so I'd like to welcome Chuck back to the academies.
Right. Thank you. Megan, that was really helpful. Maureen's, I was chair until December of Banner, and I had insight. So legislation and helping put together some of the language. So I'm going to talk a little about more focused on the soil microbiome and some of the indirect and direct effects of human health. Do I have control? It's not clicking. I mean, it's clicking, but there. Okay. So Megan already went through the legislation. I guess my observation over the years, I've been at conferences to talk about soil health and connection to human health, and everything is always about heavy metals and toxicity. They never mention the microbial component. So I saw this as an opportunity to really gain understanding of the connection between the soil microbiome and human health, and both direct and indirect, but on the positive side, not so much. Everybody talks about the negative side of soil. So again, I'm a soil scientist. The secret's in the soil, and I use some diagrams here, but really the whole concept is this soil health is part of this One Health concept, and understanding the soil microbiome, the connection with animal and human. So there's this direct connection either through aerosols, there's eating the soil, geophagy, that people, cultures will eat soil directly for multiple benefits. And then, of course, how can we harness the power of the soil microbiome? And we have some technologies now that I think allow us to open up that black box. For the non-soil microbiologists in the crowd, I always like to put this in here. There's a nanomillion of bacteria on the earth. I never heard of that until this grad student that wrote this paper with Ian Pepper and I in 2009 came up. She didn't come up with a word, but she found this word. I never heard of this. So there's 10 to the third bacteria on Earth. Bacteria or microbes have been inhabitants over 3.5 billion years. Most of you know this teaspoon of topsoil contains over 1 billion bacteria. But the key point here I've highlighted is that we've only cultured 1%. Now we can genetically identify a lot of those organisms, but we can only culture 1% of those bacteria species. So the question is, and you'll see here in the theme, what's the other 99% doing in the soil? So it all started, well, maybe not all started, but Selman Waksman, most people consider him the father of American soil microbiology. He discovered streptomyces and neomycin. He went around the country, or his students went around the country collecting soils and looking for the possibility of discovery of antibiotics. And streptomyces in 1943, I think, was discovered, and he won the Nobel Prize for that discovery. If you want a factoid, that streptomyces came from Missouri, the Sanborn Fields on the Missouri campus, which still exists. So a little factoid there. But soils, we did Ian Pepper and I and others did this review 2009, looking at soil a public health threat or savior. And one of the things that we discovered, and I'll highlight a couple things in here, is that the soil is a treasure trove of natural products. Most of those coming from microbial activity, critical to important maintenance of human health, antibiotics, but anti-cancer drugs come from those products. Other antioxidant activities, bioinsecticides for your biopesticides, and can be a source of anti-diabetic agents and immunosuppressant drugs. So in 09, I got a undergrad student that's a pre-med major want to update this review in 09. But, you know, everybody thinks, I guess I don't know if I have to use this or not, but that, you know, at the imperial jugs, 80% come from soils. But what was surprising to me was anti-cancer drugs, 60% come from soils. Prescription drugs, 40%, non-chemical products, new chemical products in 2009 was about 50%. So again, that was before we have all the technologies and the potential discovery for that. So I'm going to show a couple slides on that, or to emphasize that fact. There was an article that came out in 2012 finding that rare actinomycetes, that most of our antibiotics are coming from streptomyces, but other actinomycetes are pretty rare, but they're finding some new chemicals isolated from soils that provide an opportunity for new drug discovery. There's other, to keep it short, other antibiotics. I didn't put the year there. Sorry, at 2018, 2014. There's a reference list at the bottom. There is about 3,000 new antibiotic resistant genes discovered from an ag and grassland soils that was done in 2014. Another study on actinomycetes, 2020, a whole new class of antibiotics. And I forgot the reference there, but over 18,000 new biomolecules have been discovered through biochemical testing from soils. And that provides an opportunity not only for human health, but other industrial processes as well. So again, we don't know who those microbes are. Well, we know maybe the genes, but we don't know their function in the ecology of those microbes. So that's on the direct human health side, antibiotics, anti-cancer drugs. And again, with the new technologies and for future human health, we need to identify those products. The other thing is more on the nutrition side is, you know, we know the history of rhizobia and the nitrogen fixing in legumes, synthesis or nitrogen fixation. But now there's new rhizobia or bacteria that can fix nitrogen on corn in the rhizosphere. Pivot Bio is one commercial product right now, still being evaluated. But the possibility is that now we can replace nitrogen fertilizers and make that nitrogen more timely and potentially increase the protein content of wheat, some of our major grain crops that are non-leguminous. And so that has an environmental benefit as well as potentially a human nutritional benefit. On the phosphorus side, we have mycorrhizae in the ability to take up phosphorus more efficiently than most plants can. About 90% of all plants are mycorrhizal, and the hyphae help facilitate the phosphorus nutrition in all those plants. And I'll bring that circle back around on that topic. But there's other things there's biopesticides, biostimulants, other things that there's companies coming into place to look at biologicals rather than chemical products for enhanced plant nutrition and hopefully human nutrition as well. So again, this new discovery and new avenues for using biologicals, the soil microbiome, to enhance the nutrition of plants, but humans as well. Okay, so that's some of the potentials. I wanted to talk a little bit about the soil management side. So we're not doing very well, treating our soils very well. The national average soil erosion is 2.5 tons per acre per year. USDA NRCS has a tolerable limit, but why should soil loss be tolerable? And we're not 2.5 tons is not going to get replaced in the time frame, in a year time frame. Joel Handelsman argued that we shouldn't have any tolerable loss, right? But again, the rate of formation is 0.1 to 0.5 tons. Some regions lose soil on the order of 30, 40 tons per acre per year. And then this graph here shows the loss of carbon. This is in Kansas over cropping seasons, but we've lost about 50% of our carbon in western Kansas because of tillage. And the global average is somewhere, the global range is anywhere from 30 to 70% carbon loss. And that's the feedstock for those microbes in the soil. I use this example a lot. I need to do sit down and get another lit review done. But this shows from Argentina, a grape mollisol prairie soil in Argentina, showing the loss of nutrients in that system over 80 years. So here we can see carbon loss in that case was 35%. But what I like to highlight is these micronutrients here. Zinc here being 40% loss. Zinc is a critical element for human nutrition, for infants, for both physical and mental development. And soils in Africa are particularly degraded. And we think about nutrients for the plant, but do we think about the whole connection to the humans? And I think that's we need to draw that transdisciplinary research. We need to look at the nutrition from soil all the way up to the human development. And we don't do a very good job of that. That's a new opportunity, and we need to solve that. Coming back to mycorrhizae, we know that mycorrhizae can not only take up phosphorus, but it can take up some of those immobile nutrients like zinc, copper, and iron. So again, we need to think about the microbiome. How can it facilitate, particularly in countries in Africa that don't have the resources for fertilizer? But even in the US, if we can look at more efficient use of our nutrients, that's going to have a benefit on human health. Okay, so this is my infamous Holy Trinity of soil. If you've heard several podcasts by me. So if you had to pick one, so I do a lot of soil health assessments. If there was one thing students ask me what to pick, I would pick carbon or organic matter, because that's, again, the food for the microbiome. But if you allow me 3 assessment tools, it'd be soil organic carbon or organic matter, some measure of soil structure. We like aggregates, but infiltration, but some measure of structure, and some measure of microbial activity. I have my favorite 1 or 2, but we can talk about that. But the reason why I put that up there, the Holy Trinity, is that they're interlinked. Carbon is the food for the microbes. Microbes recycle nutrients. They also help build soil structure. Soil structure means better water and air movement and makes it more climate resilient. And soil structure that also holds carbon and carbon sequestration. And in fact, we did, okay, slide out. Okay, so if you think about then that Holy Trinity, there's several things: climate smart ag, whatever you want to call it, conservation, agriculture, regenerative ag is a common term right now, or hot term right now. But the idea is, how can you minimally disturb the soil, do cover crops, crop rotation intensity, and integrate livestock. And the socioeconomic piece here is where is it placed and acceptable, and different strategies for regenerative ag or climate smart ag, whatever you want to call it. But the idea here is then how can we maintain the soil microbiome and contribute to that holy trinity? So this is a slide showing the relationship, that three-way relationship. We have more hyphae in the soil, you get more aggregation, and more hyphae also means more carbon. We actually suppressed mycorrhizae hyphae in certain soils, native prairie soils, and we lost aggregation in an undisturbed system, and we lost carbon. So just the fact that we suppressed those hyphae without tillage, we lost structure and carbon. That amazed me. I didn't expect that in a native prairie system. We've had an opportunity to do, what's my time, Kara? Okay, so I have a master's student has done a precipitation gradient across Kansas. We go from 400 millimeters of rainfall in the western Kansas to about 1,200 millimeters in southeast Kansas. We have more variation across the state of Kansas than from Kansas to the Atlantic Ocean. So it's a perfect living laboratory to understand how climate and soils and microbes interact. What you see here is I've just picked some of her data, but we also we're blessed with having native prairie across the whole state and comparable ag lands across the whole state. And what you see here is along the precipitation gradient, the actinomycetes in those soils. Again, it's an important antibiotic production system. And you get more actinomycetes as rainfall increases. That's not surprising. And we have different depths, and we actually measure down to almost 2 meters. On the ag sites, what happened? We decoupled. We lost that effect of the precipitation gradient. It's greatly reduced. And so the question is, what's that microbial resource for nutrition and human health? In restored prairie, we recovered that. So that's interesting. The question is do we still have those genes there, or the right kind of population that's restored? We don't know that. But, we know that even in ag systems, we got a soil health network across the state of Kansas. This is just one example where we have 5 years of data, and with the introduction, they're already doing no-till, and with the introduction of cover crops, then we can increase fungal biomass, we can increase aggregation, we increase soil carbon after 5 years. I didn't expect that fast, but it did happen. And we got this now, we got 6 farms, on-farm research trials, as well as some other traditional research trials that show that. But again, how have we changed the genetic structure of that? We know we got more fungal biomass, actinomycetes, but then how is that changing that resource? Can we recover those products that might affect human health directly? Okay, so my thoughts, I hope you all consider this transdisciplinary collaboration. When I was chair of the board on ag, I always pushed, we need to have connections with human health. We need to have nutritionists. We need to have the medical profession. Because of the genetic resource, bioinformatics, ecology, you name it, connecting with soil scientists and soil microbiologists. Also, we need to have industry involved, the pharmaceutical industry and the other industry components. And I did put in here, we need to have collaboration with social scientists and economists, because if we're adopting practices, that they have to be socially acceptable, and we have to figure out the economics of those production systems as well. Summary, the soil microbiome serves as a genetic resource, not only for human health, but for ag systems and plant nutrition. We need to get the pharmaceuticals, even those genetic resources, the bioindustry, the bioeconomy, Diana, that the fermentation industry, the decomposition or fermentation of cellulosic products, biofuels, those are soil microbes, right? So what's the future of so many of those opportunities in the bioeconomy? We need to look at, and I think there's interest, but need to look at more the rhizosphere for improved plant and human nutrition. How can we manage that system, that soil and the soil microbiome that gets into the soil health and the practices? Do we know the loss of those genetic resources? If we decrease those populations, what about the genetics of those, the potentials? And can we recover those? We got higher microbial biomass, but do we have the same genetic structure? I don't know. And then my favorite thing is, I'm not a geneticist, but how do we translate those genes into ecology and function? That's a gap. So that there's some references there, and I got more, and so I can take questions.
Great. Thanks very much, Chuck. Okay, I'll now open it to the committee for questions. Mike.
Chuck, thanks for that presentation. You showed some data on, I guess it was native and restored prairie and ag, soils and different levels of microbes in the soil. I think that's what you were showing there. Could you speak a bit to, we're talking about microbes in the soil, but obviously that soil has depth, so there's spatial differences. We have different crop rotations that go on those soils. And so, we presume that those crops might, recruit and have an impact on the soil microbes. So maybe a little discussion of that, if you could.
Yeah. So again, this is a study where we looked at this precipitation gradient. The native prairie was all C4 grasses primarily. So it's pretty much the same, a little bit structure as you go from east to west to the drier climates before we get into Colorado. But the ag, yeah, it does vary. It's sorghum and wheat in the western part of the state, and corn and soybeans on the east, and then your mixed match of things across the state. So yes, so far in our work, I don't know if the crop composition rotation has as much effect as the precipitation and the total plant productivity. It's really how much carbon can you pump into that soil. Now, I suppose if you get to exotic, deep-rooted plants, perennials into that rotation, we are looking at some of these on-farm trials where they're doing perennials in the rotation, a little bit different root composition, quality and depth. Like I said, we did actually measure down to 2 meters. What scared me was the one site in western Kansas is one that I started 34 years ago. Show my age when I got to K-State. That was native prairie, and we put it into no-till wheat and sorghum. And we lost aggregate structure all the way down to over 1 meter. I did not anticipate that. I knew we would change it in the surface, but I didn't expect that. And we've lost mycorrhizal fungi, microbiomass, and that, but I didn't think we would dissolve those aggregates that fast at that depth. So now we have sites in this gradient that we all those sites were conventional ag, but we do have sites there's no-till or no-till plus cover crops. And we know that we're starting to recover microbial activity, biomass, actinomycetes, whatever, or mycorrhizal fungi in the top 15 or 30 centimeters or so, but not at depth yet. But we don't know. Some of those sites been in no-till for 20 years. Yes.
I had a question about the nitrogen fixation in corn that you
Yes.
talked about. Is that just free living nitrogen fixers or
Yeah,
is
it's
it some?
associated nitrogen fixers. It's Klebsiella, varicola, then gene edited
Oh, okay.
to knock out the regulatory gene for ammonium control.
Yeah, that's what I was
So it can
wondering.
fix nitrogen in the presence of high ammonium. It's living on the root and it's supposed to transfer that ammonium fixed into the root. But there's other companies that are doing GMOs. They have a regulatory hurdle. But I think the idea is there's potential there to look at these associated nitrogen fixers. Whether we can get a corn to be a symbiotic rhizobia, I won't be in my lifetime.
Yeah, yeah.
There's too many controls. But I like the idea, I like the approach of associated nitrogen fixer.
Yeah, I was wondering also about that bioengineering and how you think that would be received by the regenerative community, organic, if it is a really good.
That's the question. Regenerative.
That's true.
People, and we're developing potentially a regenerative ag initiative. And using natural microbes, I mean, it's gene edited. So I guess that's why we're doing the social surveys on that scientist component.
We've got one online, Lindsay.
Oh, hi. Yes, this is Lindsay. I actually wanted to ask a similar question. I was also curious to hear your perspectives on the current and potential feasibility of using direct inoculation of soil microbes, whether using like strains that are regionally or locally isolated, or commercial inoculums and things like that, or as we were just talking about, kind of the modified organism side as a strategy to increase both agricultural yield and quality and nutrient density of those products.
When I taught class 20 years ago, I used to say, forget about inoculating the soils. There's already 1 billion of them there. You're adding 100 cells per gram. They aren't going to compete. They're lab organisms. They're can't compete. They're not tough. I've changed my tune the last 10 years. I still believe these general inoculants, my opinion, are a waste of time. I tell the students send me their checks. I probably can make better use of the money. At least try to. But I really think the rhizosphere is an opportunity because it's rich in carbon. It's more like a lab environment, not necessarily, but there's more carbon there for the non-natives to survive and compete in that environment. And I think there's a whole industry out there looking at seed coating or inoculants in the rhizosphere. The general soil inoculants, I'm not a big fan of, but I think there's opportunity in the rhizosphere. Forgot what I was going to say, but anyway, yeah, I think there's an opportunity there, and it's a better environment. The thing about Klebsiella, the nitrogen fixer, is supposedly it doesn't survive in the right. Well, once the corn quits photosynthesizing or starts putting photosynthate into the grain, it dies out. It doesn't survive. That's good for the industry, but essentially good for the environment because you want to have it keep fixing nitrogen all year round.
So thanks, Chuck. I appreciate your point about the importance of soil organic carbon as the feedstock for the soil microbiome. And you highlighted practices such as cover crops and diversification with perennials and tillage changes as ways of trying to recoup some soil organic carbon levels, which don't have a magical way of reappearing. But I was wondering about, we had touched earlier as a committee earlier today about the role of plant and animal integration and the fact that, American agriculture has separated those two quite dramatically. And then we also have excreta coming from people. And so, what we've seen when we apply biosolids, I guess half of American biosolids go onto land, but then we've seen in the main case in particular, and generally the PFAS problem associated with biosolids. But there's a topic that I haven't heard coming here, which is sort of the role of biochar as giving persistent carbon, which could be from pyrolysis of biosolids, which would eliminate the PFAS and give us a channel for bringing some persistent carbon back into soils. What do you think of that or other means to maintain what you point out as the engine of the microbial activity and the soil structure and everything else? What options do we have?
Yeah, good question. So, well, I'll give you two components. One is we got a study where we're adding 34 years. It's classic. It's only supposed to be 3 years. Actually, I can't stop it because it's historic. I talk all day about it. So yeah, it's 34 years, no-till and conventional. Things. So my thought is much and how ways that you can get carbon in the soil is perennial. You know, I would love to have plant breeders breed for roots, depth and quality. Get as much carbon in that system. My opinion, I'm not a big fan of biochar. If you pencil it out, how much biochar you have to put on to boost the carbon levels, and figure out how much transportation costs and energy costs to move that biochar carbon onto a 1,000-acre field. And if you want to, and tillage, is not a good thing. And it's so fine, you no-till and put it on in a Kansas 40-mile-an-hour wind. It's here in DC right now. Right? So, yeah, huh? Yeah, right. If you pencil out how much carbon to really make a difference. Now, I think in certain situations, strip mine soil, surface mine lands, farms, degraded lands, where you don't have zero or little carbon, where you can make a big difference. But I just think in productive mollisols that I wouldn't see it as a mechanism for a big boost in carbon, if that's what you're trying. Now there's some other traits like binding other things, metals and that do compost or other ways to get carbon. My opinion.
Thanks. We're going to have to close it here. Chuck, thank you very much. I appreciate you coming and presenting. We're going to next hear from Dr. Wayne Honeycutt. He is the president and CEO of the Soil Health Institute, which works to safeguard and enhance the vitality and productivity of soils. He previously served for 5 years as the deputy chief for science and technology. Sorry, I keep giving everybody different titles. With the Natural Resource Conservation Service of the USDA. He was also a research soil scientist for 14 years and a research leader for 10 years with the New England USDA Agricultural Research Service, New England Plant, Soil, and Water Laboratory, where he led and conducted interdisciplinary research on carbon, nitrogen, phosphorus cycling, and sustainable cropping. Last year, Dr. Honeycutt, and his co-authors reviewed the literature available on links between soil health and food nutritional quality, which is part of what we're very interested in. And he is going to share some of those findings with us right now. Dr. Honeycutt, thanks so much. Appreciate it.
Thank you all very much for the opportunity. Can you see my screen all right?
We sure can.
Absolutely. Good. Well, thanks again for the chance to visit with you all today. I first need to acknowledge that I'm the third author on this work. Dana Bourne was a master's student and working at the Friedman School of Nutrition Science and Policy at Tufts University. And I brought her on as an intern just for a summer. And she and her advisor and I worked with her all summer. We met with her about weekly on her progress. But this is really her work that I'm going to talk to you a bit about. Tim Griffin was her advisor. He was at the Times associate professor at Tufts in that same school and is now a dean there. So I just wanted to acknowledge that this is their work, and I just am the beneficiary of being able to present it to you today in many respects. I believe I need to go to presentation mode, Donna. There we go. This will help. All right. So it's already kind of been mentioned, Megan mentioned, and
we're seeing your presenter mode rather than the.
Oh, you are? Okay.
If you press the three dots there, yep. Hide presenter view. Yes.
All
Great.
set. Thank
yes.
you for telling me. It didn't appear that way on my large monitor, only on my Surface next to it. So thank you. So it's already been mentioned, that's a conference that we held in 2018, but I also want to acknowledge that the National Academy also had a somewhat similarly titled conference, I believe it was in 2016, or at least a study starting to explore some of these relationships. This particular conference that we had we had about 200 folks, and probably about 140 of them were ones that you would expect to be present for kind of a soil related conference. A lot of agronomists, soil scientists, but about 60 of them were more in the public health arena in one form or another. And there's it's just kind of just wanted to show you just kind of the outline of some of the things we worked on for a day there with this group, with this workshop. Hearing from the medical community, their perspective on soil health, human health connections, the soil science community hearing the same thing, starting to address those connections between soil health and human nutrition, soil health food safety, the biotoxins, interconnections of food, human microbiome with soil, implications of soil health with human populations and communities, started looking at some of the policies and their intersections and their when they were connected and when they're disconnected. And then we really then talked also about a number of funding opportunities and challenges that we could see. And out of the 10 recommendations that came from that, about 5 of them really touched in one way or another on the relationship between soil health and the nutritional quality of the food that people eat. And so I wanted to see if there is a way that we could just start to explore this relationship. Being a nonprofit, many times we have to kind of start things like this on a shoestring. And sometimes we don't even have that shoestring to start something on. And so our premise here was that there are enough number of attributes of soil health that do lend some feasibility to the premise that enhancing soil health can lead to greater nutrient uptake and therefore nutritional quality of the food grown. And so this was, back 2 or 3 years ago. But since then, we've conducted one particular study that I just wanted to share with you some of the results that really kind of lends credence to that premise. This particular project was one in which we evaluated over 30 different ways of measuring soil health, 30 different soil health indicators, at 124 long-term agriculture research sites across the US, Canada, Mexico. So you can imagine a wide range of climates, soils, production systems, management practices that were represented. And each of those sites, there was some type of conventional system compared with some type of soil health improving system, generally in a well-replicated design. So they had the appropriate experimental design to allow us to come to the appropriate statistical conclusion at how effective each of these measurements was. And just so that these are just some of the results I wanted to share, just again to kind of backstop of that premise. What we're looking at here is a number of different indicators, the mean shown by the dot and the 95% confidence limits shown by the bars around that dot. In this particular one, I'm looking at the response to decreased tillage. So we had like 51 sites out of those 124 where they were comparing some form of conventional or baseline tillage with some form of decreased tillage, whether it's moldboard plow to reduce till, or moldboard plow to no-till, or reduced till like chisel plow to a no-till, but some form of reduced tillage or decreased tillage. And so the interpretation here is across those 51 sites, 161 treatment pairs, on average, there was about 12% higher. The total nitrogen in soil was about 12% higher on those soils with decreased tillage than for the soils that had higher rates of tillage. And so what you can already see is that the total nitrogen was higher in those soils. We're using that soil health promoting practice of reducing tillage. And in some cases, there were indications that not only was the total nitrogen available, but more was there, but more of it was also available. Potentially mineralizable nitrogen does not stand up because that 95% line covers the 0 point here. But this autoclave citrate extractable protein does hold up. This N-acetyl beta glucosaminidase is the enzyme that's involved in breaking down chitin. Both of those are really kind of implicated in indicating that that the nitrogen is more plant available, more easily cycled. And so you can think of greater nutrient availability, but then also there's some structural considerations here, like Chuck was talking about his shock and dismay at seeing aggregate stability down at deep depths and the importance of aggregate stability. And so as a kind of a good measure of soil structure. And so when you think of improved soil structure, now you can think of how easily the roots can travel through that soil. In a similar way with bulk density, if you have a lower density, you can think of how easily now the roots can travel through that soil to take up water and to take up nutrients. And then available water holding capacity was also significantly higher when you reduce tillage. So now when you get a heavy rain, you have now more of a chance to store more of that water. So now when the nutrients can move by, whether it's by mass flow or by diffusion, to those plants, to those plant roots, then now you would have to think that what we're looking at here is really an overall picture of more nutrients being available in the soil health promoting system, like that has reduced tillage, but also that the physical structure of the soil is more amenable to the uptake of those nutrients. And now you can hold more water in that soil, more of its plant available. And so again, so now those nutrients can go to that plant through that water. And so there's just kind of the whole situation does set up to where it just makes sense to us that we should be able to increase the nutrient uptake and availability because of that greater availability in the soil, greater moisture in the soil. We should be able to increase that nutrient uptake by the plants, and hopefully that would show up in the food that's grown on those plants. Similar types of relationships. This was not tillage. This is with cover crops. We only had 21 treatment pairs, but again, in many cases, that response to cover crops, that's another soil health improving practice, showed that there should be, that the premise essentially really kind of stands up. And so given that premise, our question basically was, can we reassess the studies in the published literature through the new lens that will either, confirm or refute that relationship? And, it's kind of, I guess, a wordy way of saying that when you're a nonprofit and you don't have the funding for doing a study like this, obviously what you would want to do, you would want to set up experiments where you can control all these different things, that you need to control so that you can manage the error, the variability, so that if you're going to detect a difference, it's a true difference. And if you can't detect it, you want to know that it's because it didn't exist. It's not because you had so much variability. And but we were not in that position. And so what we did was basically is something that had been eating at me for a couple of years, is thinking that, there are so many of these studies that have been published for decades where people have looked at things like reduced tillage on things like yield or cover crop effects on yield. And it just made me feel like, are there enough of these types of studies that not only have just looked at yield, but that also have documented changes in the soil and that have also documented things like a nutrient uptake and perhaps nutritional quality of the food that's grown. And so that was the primary question is there enough in the literature to really kind of assess this? And so our approach was just to conduct a literature review. I want to point out it's not a meta-analysis. We were not taking all the data from all these different studies, putting them into one database and analyzing like it was one study. It was not that type of analysis. It was just basically looking at these literature papers, or papers in the literature, I should say, one-on-one. And so that was the task of our intern for that particular summer. And so ideally, you know, what she started looking for, and we visited with her on what those kind of ideal papers would be. And we, of course, looked at hoping to find papers where that compared the different management practices on these soil properties and on these crop properties that did provide some measurements of soil health under different management conditions. That also was measuring some of the nutritional aspects of the crop and what would become food from that crop. And we realized that in many cases that, again, because these papers, these studies were not designed for thinking about human health. They were designed for things like, you know, what's the effect of reducing tillage on yield? And so, but we were hoping to at least find some that also at least considered or discussed, if not measured, those relationships to human health. And her initial assessment, you know, anytime you do just kind of a keyword search, you often end up with thousands and thousands of papers. And when you start to boil them down, you realize it's just a fraction of them that are applicable. She basically kind of needed to focus in on something. And so we had her focus in on wheat and tomato. And that's, of course, because of, you know, they're both nutritionally important, they're economically important, they're agronomically important in agriculture. And they're very different. And so that was the focus. I'll just tell you right now, just kind of, I'm going to show you some examples, but the bottom line is that there was so much inconsistency it seemed like that it was so difficult to have all those criteria met by a single study. There were just so few and far between that largely the results are inconclusive. And so it's always hard to say that, but that was the conclusion that we reached. But just to give you kind of some flavor, some of the examples of what she found. Here we have two different soils. This is Australia, what they refer to as a Candesol, looking at conventional tillage and no tillage, just looking at the grain yield in wheat. And here in this particular one, they did have found a higher percent of protein in the no-till situation. But then in a different soil, you can see just the vertisol, it's kind of a high shrink swell type of very clay soil usually. It was the very opposite. Again, the no-till was different than the conventional till in terms of the protein, or that I think when I think of this, I'm thinking of kind of nutrient density is when I'm thinking of the concentration of protein in the grain. And so you know, this is one of the realities, is that the same study done the same way, but two different soils, they found two different effects, two different impacts, both increase and both decrease. And so basically that kind of tells me at a minimum, we also need to think about those inherent soil properties when we are exploring these relationships and the interaction of the management practice on the soil. Here in Saskatchewan, this was one that was not so much like a tillage type of comparison, but it was more of a rotation. Of course, rotation can be an important component of soil health also. Looking at continuous wheat, versus some different combinations of pea and wheat and canola pea and wheat and canola wheat. One of the things that that she found, or that they reported, was significant increases in their grain yield compared to the continuous wheat. And it's a similar way with the total content, multiplying basically the concentration by the yield. Now, I apologize because it seems to me that if these are all significant and these are all significant, then there's a good chance that these were also, but the table in her report, as I was looking at it last night, I did not find those, and I could not find that article to verify one way or the other. But what I could find, did have access to online, was their abstract, and their abstract stated that the fall residual soil nitrate and available water accounted for 12 to 24% of the increased grain crude protein concentration in 3 out of 5 years. So again, it's a situation that's not every year, but in 3 out of 5, to me, what that is telling me is that the relationship to the soil properties, in this case, the nitrate and probably the available water holding capacity, says available water, was related to that nutrient density, or that protein concentration in the grain. Here's another example over in Italy. What we have here is data from 3 different years, comparing the conventional tillage and the no tillage. And here's a situation where they reported in 1 year that the no-till, which is a soil health, again, improving practice, did have a higher concentration of protein in the grain. But guess what? In the following years, it did not. It was significantly lower. And then again, in the following year, again, it was significantly lower. And of course, some of this can interact with the local weather. You got to figure that this was a very dry year because their yields were so low, relatively low in that year 7. And so, again, this is part of the challenge of trying to look at interpreting studies that were designed for one cause to try to see if we can open up the door to insights that could be gleaned from looking at the data, trying to analyze the data from a different perspective with a different purpose. But this is the variability that they found. This is the only example, I guess, that I would provide with tomatoes. There was less quantification. Most all of the tomato comparisons were conventional versus organic. This one particular study, the only thing that I saw was they said the soil organic matter was higher in the organic system, and they did have higher flavonoids. These 3 that they listed here in the tomato fruit than they found in the conventionally grown tomatoes. But again, this is a part of that challenge that we have with looking at the data in this way, is trying to have enough soil information also to go along with it. So just a quick summary is that the results were inconsistent. One of the challenges, of course, that there are very few of the studies reported that sufficient data that really gave us that breadth of crop and soil management information we needed, the soil measurements, the nutritional quality components that we felt like, are really needed for really thoroughly assessing it. But I feel like it's really important that it's also acknowledged that we are not criticizing those studies, because, we were trying to use those studies that were designed for one purpose to see if we could answer another question with them. So this was our attempt at doing that. And I guess I would also say that this does not mean that soil health and food nutritional quality are not related. I think it only means we were not able to consistently find it with this data for the reasons that I just described. So there were a number of recommendations that I hope may be helpful. It's kind of interesting. It sounds like some of them already been talked about today. We referred to interdisciplinary teams here, and people have referred to transdisciplinary. I think there's obviously a lot of connections to those, but they need to really be well designed and really somewhat standardized. Just for example, no-till means never till to some people, but it means I till 1 year out of every 3 to other people. And so there are just things like that that really need to be standardized in conducting these types of studies. In a similar way that really need to have standardized measurements for soil health and soil properties, crop growth, and all those contents. And so I have just a couple more minutes. There were some additional insights that I felt like might be helpful to this committee, a bit off topic from what you all asked me to cover or that I just covered. But there were some insights from our conference that I just wanted to share because I thought it might be helpful to you all. So if you'll allow me just another couple minutes to do that.
Sure, go ahead.
Thank you. One of the things that struck me, and I'm sure it probably does not strike many of you all from what I've been hearing in the last couple of hours, but I was struck that that conference that so many in the medical community that were present really large viewed soil as a contaminant. And I kind of had to grab the microphone at the very end of that workshop day and say, wait a minute, the soil's not all bad. You know, and it's the giver of life, not just the taker of life. And but there, I learned that from that conference, I mean, there's really, really good reasons. And I know from hearing the people on the committee go around the room talking about their expertise that I don't need to be saying all this. But there's, I think a lot of it is things like with lead, and now we're hearing PFAS and other contaminants. Here's some of the data like that they're looking at is the lead concentration in blood on the y-axis versus the lead concentration in soil on the x-axis. And the fact that over this one particular survey, over 500,000 children ages 1 to 5 had lead concentrations in their blood exceeding 50 parts per billion. And basically that study was saying that there's no level of lead in the blood that's considered acceptable. Anything is unacceptable at that point. And it's kind of interesting. I also learned from some of that there have been a lot of studies that not just in children, the impact that things like lead and soil have in children when they're playing in the soil and they get exposed to it, you know, from when we had leaded gasoline and paints and stuff like that. But that the elevated bone and blood levels are related to are correlated with aggression, destructive delinquent behavior, attention deficit, hyperactivity disorder, criminal behavior. And so there's definitely adult behaviors too that can be impacted by it. Now, and so I just felt like I wanted to share that perspective because you all will, you know, have this this goal of putting this package together of soil health, human health relationships. And so I wanted to make sure that you knew of kind of that perspective that existed. With the medical profession, but also, and equally, I wanted you to know about the advocacy opportunity with the medical profession too. I learned about 5 years ago of this Medical Society Consortium on Climate and Health, and they just started in 2017. But I just looked it up yesterday, and now they're up to 46 national medical societies that are members of it. And some over 70% of the US doctors are represented. So these are societies like American Pediatric Association and things like that. And so, but they are very active. They were actually, I just read one of their a post in March. They came to DC and visited with some 60 congressional folks on some of their goals, basically for addressing climate change. because they see that as really kind of the number one threat. And then also want to make sure you all are aware of this organization. I had never heard of it, International Medical Geology Association, that concerns, how geologic materials and earth processes affect human health. So I thought that might be something that you will also want to look into. And then of course, the resources that have already been mentioned, the conference report from 2018, and then the relationships that I just kind of summarized. I may have already scared you off of wanting to see them, but I wanted to be upfront with you on what they showed and what they didn't show. So thank you very much. I will stop there and see if you have any questions for me.
Thank you very much, Wayne. I'll now open the floor to the committee for any questions, comments.
I wish I could have given you all the right positive answers on that literature review.
We wish you could have too.
But we're so glad you did it.
Well, Kate, we appreciate you serving on our scientific review committee for the Institute.
Okay, Joe has a question. Joe?
Yeah, I'm just curious. The studies that you showed kind of mixed up connection between the soil health and protein content, but it seems you're just looking at no-till versus till. Was there any way any work to confirm whether the no-till was actually beneficial to the soils?
Well, that was part of the issue, part of the problem. That was our goal, was to be able to make those connections, but many of the studies did not provide that type level of information. Some of them did. In some cases, yes, it was. They were able to show that they were improving soil health, or I would say indicators that we think are important, like Chuck mentioned, like organic carbon, aggregate stability, water holding capacity, those types of things. And so there were some studies that did that, others did not.
Thanks very much. Any more questions? Wayne, thanks very much for bringing this to us.
My pleasure. I wish I could be bringing you more, but thank you for the opportunity.
Well, I appreciate seeing the breadth of what you've done. I think that helps us. Okay, next we're going to move on to our final presenter today, Dr. Maul Day from South Dakota State University. Dr. Day is a professor of nutrition science, as you can see on the screen, in the School of Health and Consumer Sciences, and the founding director of the Molecular Nutrition Research Program. A major focus of her research has been nutrition trials in upper Midwestern adult and older adult populations to understand the diet microbiome interactions and how they impact chronic disease risk and health span. She's done controlled feeding trials, which have evaluated the USDA recommended dietary pattern with different protein sources like red meat, poultry, lentils, a calorie-restricted weight loss plan, and a resistant starch-enriched ethnic diet. Dr. Day, we're looking forward to your presentation.
Thank you, Dr. Wall. Can you hear me all right?
Yes. We hear you.
Yeah, okay. All right. Good afternoon, everyone. So as Dr. Wall just said, my group has been focusing on the human gut microbiome diet connection for the past 10 years. And I'll share some of those findings with you today in the context of this committee's priorities. So we use human nutrition trials as well as translational model studies to understand how human diet interacts at the molecular level to contribute to chronic disease risk. So while we use a variety of omic tools in today's talk, I will focus on the metagenomics and the metabolomic sides of our studies that we utilize to understand gut microbiota composition and function respectively. I will use two abbreviations frequently. RCT stands for Randomized Controlled Trial, and DGA stands for Dietary Guidelines for Americans. That's the recommended dietary pattern from USDA that comes out every 5 years. So So why we need to care about dietary patterns in the context of human health? In comparison to 100 years ago, people on average now live 25 to 30 additional years. However, an increase in lifespan did not coincide with an increase in health span. 60% of the adult population suffer from one or more chronic diseases, and the prevalence increases with age, meaning health-related quality of life is compromised during those additional years of life that we have gained. The good news is that many chronic conditions can be prevented and or delayed with healthy lifestyle. However, despite knowing this, the chronic disease crisis is only getting worse. So what may come as a surprise is that way more research dollars are spent on evaluating pharmaceuticals, dietary supplements, and dietary bioactives as compared to reliably evaluating dietary patterns. That is, the robust RCT study design as utilized for drug and supplement trials are rarely used to evaluate dietary patterns. Of note here is that RCT trials become much more logistically challenging for clinical nutrition research due to the need for controlling all aspects of the diet, that is, controlled feeding studies. Hypothesis generating observational and epidemiologic data suggests that various components within a dietary pattern may work synergistically to benefit health, which must be confirmed by RCT trials. So RCT trials are the gold standard for establishing health benefits of any agent, including dietary patterns in humans. On this slide, I have listed 4 of our RCT trials in which these are registered clinical trials in which we tightly control food intake. We reported the first intervention trial of an ethnic habitual Hutterite dietary pattern, and we were the 3rd research group to ever assess the recommended dietary pattern, the DGA, in a controlled feeding trial. And this is despite that the USDA guidelines had been around since 1980. So I'll show you the results from these 2 trials in today's talk. The other 2 trials listed are currently running, evaluating the effects of DGA on risk factors for diabetes, heart disease, sarcopenia, and Alzheimer's. These conditions are among major chronic diseases listed by CDC on the bottom panel. In all of our DGA trials, we designed the diet based on the upper Midwestern palate and provide all meals, drinks, and snacks to our study participants to minimize variability in dietary intakes and to ensure rigorous data collection. Tightly controlled feeding to avoid dietary variations is critical for studying changes in gut microbiota composition and function, which intricately relate to various intestinal and extraintestinal diseases, as shown by the tree. Human body is more microbes than human, with estimated 10 times more microbial cells and 100 times more unique microbial genes than our own. Therefore, improving human health through dietary modulation of the microbiome is an evolving and promising strategy that we all know about. We are the first group to report a red meat and a white meat investigation using a DGA diet. We compared how intake of chicken and pork affects cardiovascular risk using a unique non-inferiority trial design. Heart diseases have been the leading cause of death for more than 10 years in a row and have a large toll on the economy. Trimethylamine oxide, or TMAO, this molecule here, in short, has emerged as a newer independent marker for atherosclerosis. TMAO is a co-metabolite resulting from the host and bacterial enzyme activities. L-carnitine, choline, and betaine are the main dietary precursors of TMAO. These molecules are typically higher in animal origin food, especially L-carnitine being higher in red meat, which is why some studies promoted that red meat intake induces higher TMAO production than white meat. Intestinal metabolism of these molecules by human gut microbiome produces trimethylamine, TMA, which is shown here in the middle. So this is exclusively produced by gut microbiome. And then TMA then enters the liver through the portal vein circulation. It is then oxidized to TMAO by an enzyme called flavin monoxygenase 3 in the liver. Studies have conclusively shown that animal protein induced higher TMAO than plant proteins, but data was less consistent regarding TMAO response with respect to white or red meat in omnivores. Pork is the most popular animal protein worldwide and third most popular animal protein in the US. Thus, scientific clarification regarding its effect on TMAO is essential for omnivorous consumers to make informed dietary protein choices. Pork intake was previously never evaluated in a control feeding study for TMAO response. So the research questions we asked were if lean pork intake differentially modifies TMAO compared to poultry in omnivores. Our scientific premise was that the omnivorous gut may not differentiate the two animal proteins. While many nutrition studies extrapolate negative results as non-inferiority, statistically speaking, negative results cannot establish this. The non-inferiority trial is a rather unique approach for nutrition research, where placebo control is replaced by active control. Here we use poultry as the active control, as it is widely recommended, including by American Heart Association, as the heart healthier animal protein. For secondary outcomes, we explored if pork intake as part of a DGA diet influences human gut microbiota, TMAO substrates, a panel of 346 biogenic amines, lipid profile, and oxidative stress markers. All study meals and snacks were prepared in our metabolic kitchen laboratory. 156 grams of poultry or pork per day provided more than 60% of daily protein to healthy study participants 50 years and older. The table shows no difference in traditional lipid markers, oxidative stress markers, and kidney function markers, and the compliance to study meals and the study protocol was very high. Individual paired dot plots with mean and standard deviation indicate that the participant had similar TMAO levels after pork and poultry intake. But statistically speaking, the absence of difference is not a similarities. So we carried out non-inferiority testing as shown on the right. We see that the response of both circulating TMAO and TMA again is the microbiota-mediated intermediate metabolite, to pork was non-inferior to poultry at 97.5% confidence interval. Since the means were below zero and the 97.5% confidence interval was did not cross this boundary here, which is the non-inferiority margin, we can interpret that pork intake did not increase the TMAO and TMA response compared to chicken. Similarly, the relative abundance of dietary TMAO substrates, betaine, choline, and L-carnitine were not different between the two groups, as you can see with the graph on the left. Furthermore, pork did not alter the overall biogenic amine signature consisting of 346 different metabolites. The data are not shown on the slide. It's a lot to show. These metabolomics outcomes support our observation of pork being non-inferior to poultry. Alpha and beta diversities of microbiota at the phylum, family, genus, and OTU level were not different between the diets. On the left, principal coordinate analysis with Jaccard index shows that the 95% confidence interval boundaries for both chicken and pork have shifted in the same manner, differentiating from the baseline. This indicates a clear impact of DGA on the gut microbiome independent of the two dietary protein sources. Of note, the baseline diet was significantly higher in fat and sodium than the dietary guidelines diet. On the right, the stacked bars show comparable shifts in the abundance of gut microbiota phylum between the pre and post diets in pork and chicken groups. Thus, we conclude that the gut microbiota is minimally influenced by pork compared to poultry in omnivores. During our analysis, we noticed individuals had a substantial variability in their baseline TMAO levels, even though the participants were consuming similar macronutrients during the run-in phase. The diet-induced TMAO change was strongly associated with the baseline levels, independent of the protein source, as shown in the figure, with near parallel regression lines. To follow up on this observation, we are running an additional analysis of blood and urine metabolites. From what we are seeing thus far, it appears that DGA has a beneficial effect on TMAO metabolism, irrespective of dietary protein source. Unlike extrapolating negative results, our study presents non-inferiority testing-based evidence. We show for the first time that consuming pork as a predominant protein within an omnivorous guideline-based diet does not exacerbate TMAO, its precursors, biogenic amine metabolites, and has a minimal impact on microbiota features. Pork also had a non-differential impact on lipid panels and oxidative stress markers. Overall, there was a minimal impact of animal protein substitution on microbial metabolic output in omnivorous consumers. This suggests that lean pork may be incorporated as a primary protein within a guidelines-based diet without an adverse impact on TMAO-specific atherosclerosis risk. Importantly, 95% of Americans being omnivores, lean pork increases dietary protein options. And as we know, the dietary guidelines, the compliance to dietary guidelines among the general population is very low. Having more protein choices may help improve adherence to recommendations for improved health and well-being. We also worked and published numerous results between 2014 and 2022 regarding how a resistant starch or RS-enriched habitual hutterite dietary pattern made a difference in metabolic syndrome status in hutterite population. And I will share only some highlights. I won't get into the details here. The Institute of Medicine classifies RS under dietary fiber as it resists digestion in the small intestine. There is currently no specific recommendation for RS intake in the US, but CISRO in Australia suggests 20 grams per day of RS, which is 4 times higher than what a typical Western diet provides. The US diets are also low in dietary fiber. At the time we started this project back in 2012, there was a limited number of research on RS. We wanted to examine if and how RS modulates gut microbiota produce short-chain fatty acids and immunometabolic health in human subjects with metabolic syndrome. We used 30% volume by volume RS source from wheat for intervention. Foods were prepared with RS supplemented flour. Participants could not distinguish between control and RS flours due to neutral color and taste. Hence, the study could be double-blinded. The study was carried out in hutterite colonies where the prevalence of metabolic syndrome was even higher than in the general population. They practiced communal living with central dining, meal planning, cooking, and very uniform dining schedule. As a result, feeding was controlled precisely. Habitual hutterite diet is similar to the Western high-fat, high-carbohydrate dietary pattern, with the exception that they use fewer ultra-processed foods. We did not restrict calorie intake during the study. The control arm of the study consumed a habitual diet without RS supplementation. So when RS was added to their diet, which apparently is not very healthy, They still lost fat mass, but gained lean mass compared to the isocaloric control diet. We used DEXA scanning for body composition measurements. Waist circumference was also reduced, indicating reduced central obesity. We showed for the first time RS-induced improvements in blood lipids, notably lowering of total cholesterol, LDL cholesterol, non-HDL cholesterol. Non-HDL cholesterol is considered a biomarker for atherogenic dyslipidemia. Immune function markers were also improved. All observed clinical changes were associated with the changes in the gut microbiota composition. Gut microbiota functional changes were indicated by increased short-chain fatty acid production and changes in secondary bile acid composition. The species that clustered together to associate with clinical outcomes had a higher correlation with each other, indicating a possibility of bacterial cross-feeding interactions and co-occurrence. We also followed up on the clinical trial findings for mechanistic validation using mouse model research, which helped conclude that RS-induced butyrate attenuates metabolic inflammation through epigenetic downregulation of a pro-inflammatory transcription factor. Overall, the study, it's multiple smaller studies under the umbrella of a bigger study. It shows that when a complete makeover of a habitual dietary pattern is not possible, as in case of the Hutterite with unique cultural practices, there are still ways to make subtle changes in one's lifestyle to improve well-being. Finally, I wanted to share some thoughts about the soil microbiome and human microbiome connection. We heard a variety of important and interesting perspectives already. I'll try to reflect on the topic from a little different angle. We heard about nutritional differences between, depending on different soil conditions and perhaps between organic and conventional crops, which is very important. However, what interests me is that organic farming reduces exposure to harmful chemicals, and I'll tell you why that is important from the human gut microbiome perspective, and can also increase soil biodiversity. So research has shown that numerous gut species in the human microbiome have become extinct. Ultra-processed, low-fiber diets and antibiotic overuse and modern hygiene practices in industrial nations are among major drivers. This is from a published study in Nature. It is widely hypothesized that shrinking gut diversity may have contributed to the chronic disease epidemic that we are experiencing right now. Recent research with the hunter-gatherer community, the Hadza community in Tanzania, and this was also a published study in Science from 2017, validates this hypothesis that the shrinking gut diversity is contributing to this chronic disease epidemic. So the question I wanted to, that we need to think about is that eating right, what we nutrition researchers do, that we tell people, okay, eat right, eat less processed food, eat more fiber. I mean, that's great, and that may help preserve the good species that are still around. But how do we increase our gut diversity? Is there any way we can get back some of the lost species in our gut? Acquiring it from environment, that is soil, water, is one potential option. Some case studies have shown that this is possible, for example, by growing vegetables organically and consuming them raw without much washing at all, like, just rub off the dirt out of it and consuming it. And there is a case study that showed that it increased alpha diversity in the gut microbiome. So we have to remember that from evolutionary standpoint, excuse me, the soil microbiome and the human microbiome co-evolved at a certain point in time. Hunter-gatherers were closer to the soil and nature. We now know that the Hadza gut has much higher alpha diversity than ours. Thus, the potential link between soil and the human microbiome is it's definitely worth exploring. We don't know a whole lot about the connection part. We know soil microbiome, we know the human microbiome, but the connection therein needs to be explored and researched much more. And that's something that I think is very important. With that, I would like to acknowledge my group members, researchers in my lab, and also thank the funding agencies who support our work. Thank you, and I'll be happy to answer questions.
Thank you, Maul. That was really very interesting, and I'll now open the floor to the committee to ask any questions. Yes, Carlotta.
Thank you, Dr. Day. Very interesting presentation. I think your examples on the Hutterites and the Hadza communities bring up the additional consideration of how we see vast disparities in disease prevalence and nutrition and the microbiome and how these may be interrelated. So do you have any thoughts or recommendations on how we can consider those disparities that we see, you know, how we may need to consider them in a population specific way?
So, you know, that is what, and thank you, that's a great question. As I was just saying, you know, I don't understand, and this is a big discussion in scientific literature as well, that there is, you know, why we don't do a lot of randomized controlled trials to understand how diet and their food can impact human health, right? Using food as medicine, that kind of concept. We don't have a lot of that. And then when we get there, we also need to understand how different populations and the populations in terms of different communities, like you are saying, Hutterites or ethnic communities, and then Hadza, but also the communities here, you know, different ethnically different communities practicing different, having different cultural practices or different socioeconomic populations and different age groups, right? And there is so much unknown there. And all we know, nutrition research, like 80% of nutrition research data that is available over the last 40 years, 80% of that is observational. And observational research is great. It's very important. And we need to have that to generate hypotheses. But once the hypothesis is generated, we need to either confirm or refute that hypothesis. And that is where nutritional health research is really lagging behind. So, yeah, to answer your question, we need to focus on different communities and populations, but and there is a lot of interest in that area too. It's just that it hasn't happened yet.
Kelly.
Hi, great talk. I have a question, and it's kind of linking this soil microbiome to gut microbiome, which I realize we all talked about earlier today is kind of limited in terms of its data. But when we think about it in terms of fundamental understanding, I think you gave a nice example of organic soils, and maybe organic soils would have food that was higher in nutritional quality or bioavailability, and then that could augment the microbiome for desirable features. I guess, were you thinking that, and this is something we've been struggling with in committee, is, you know, transferability of microbiomes, or were you thinking of it as like an indirect augment of the microbiome through nutritional quality of the food to impact human health? I guess I'm trying to make the pathways between soil health and human health via the microbiome and try to kind of articulate that space a little bit better. And maybe you've thought about this more and could have some insight in there. Does that make any sense?
That's a
question. So I'm sorry.
No, that's a great question. And I think there is both of those potential exist, you know, having more microbes in the soil, like, you know, doing more organic farming, crop rotation and those things, you know, that's not my expertise, but I do know a little bit about that. Those things can improve nutritional qualities. as such, and also reducing use of agrochemicals can improve soil biodiversity. Now, what I was trying to say is that, the human gut, especially the Western human gut, is really, shrinking. It's the alpha diversity is shrinking. Now, how do we recover those species that we have lost? And we may never recover the species that we have lost, but at least how do we increase our alpha diversity? So one source is the soil, one source is the environment around us. And if we have, if we can ensure the produce we consume is free of agrochemicals, harmful agrochemicals, and we can just very quickly brush off the dirt and then not rip off those produce from the soil microbes, and maybe those microbes, and again, I was making that point, and I don't know for sure, this is what is my kind of informed opinion, is that the human microbiome co-evolved at certain point in history with the soil microbiome. Of course, soil microbiome evolved much earlier. So there might be a connection there. I'm pretty sure, and again, we don't know, no studies that I know that has actually tried to compare the flora, the gut flora with the soil flora, but there will be at least several families of microbes that will be similar. So probably that could be a potential source that we can try to acquire in our gut. And it's just not like it won't happen tomorrow, and there needs to be a more concerted knowledge and effort there, but it's
I agree with you.
Yeah,
and part of our task is to find research areas that we need data for. So I think that you've articulated that nicely. So thank you.
Yeah,
I have a quick follow-up question on that. It's Kate Scow, I'm a soil scientist, but I'm often thinking about the exposure pathway of the microbiome into the human, and like how much would directly eating the soil on crops that haven't been as washed and that are safe, how likely would they wind up in the human, or how much do we know about that? Because I think of some of these studies of greater, like immunity in people who live in rural settings, but it may be that they're exposed to the benefits of the microbiome or the microbiome itself more from direct contact with the soil or being around the soil all the time, and inhalation being a pathway or dermal contact being a pathway. I'm sort of obsessed with this pathway from the soil into the human. And I wonder if you have any thoughts about that, or how much we know about the human microbiome being affected by exposure.
Sure. There is a fantastic team in Stanford University, Justin Sonnenberg and his wife, their duo team, Erika Sonnenberg. They are the one who actually goes to Tanzania and research this Hadza community. So the reference I cited on my slide is from their group. And if you read, there is a series of publications on their research, and they're researching the gut of the Hadza community and how different the diversity and the whole spectrum of the microbes in their gut versus the Western gut. And they're also reporting alongside that the practices, the cultural practices. Of course, these are hunter-gatherer people. They don't have anything even nearly as close as our hygiene standard, right? So they just pluck things from plants and hunt animals, and they just hardly cook them. It's just roasted in fire, and so it's pretty if you can imagine. So they are obviously they're completely cut off from that modern society. So where is their diversity, which is way more diverse than the Western gut is coming from? So that's the assumption there, is that they're not washing, sterilizing anything when that they are putting in their mouth, and probably it's coming from their environment. Now, there is a very nice talk that's available in the YouTube. If you're interested, Dr. Erika Sonnenberg talks about doing a study with her husband, Dr. Justin Sonnenberg, where he initially profiled his microbiome, and then he started eating all these fresh produce from his own backyard that are not washed, and then reprofiled his microbiome, and he could see that but it's just 1%, so it's not a rigorous scientific data, but it did improve the alpha diversity in his gut. So, and I do know several other colleagues who did similar type of experiments. So, but again, this is not published scientific data. These are more anecdotal evidence. But the Hudson community data is it's published. It's more concerted effort, and there perhaps I don't know that there are other hunter-gatherer communities, and even this community is very small. They're probably the last hunter-gatherer community on Earth. So what we can learn from them is so invaluable. So yes, I think funding in this area is very important. Funding in overall nutritional health is more funding is needed. There is so much funding for pharmaceutical research, not as much for nutrition research, which is a problem that nutrition researchers talk about frequently. So just something to, I wanted to share.
Thank you very much.
Thank you very much. I think what we're going to do now is have the panel. We've got two people, Dr. O'Rourke, Dr. Rice, sit at front, and then we will also have our other two speakers, and the committee will have a broader base to ask questions to. What we're trying to do here is to hear a scope of broadening our understanding of the statement of task, of what's going to go in the report, and to hear from you all is really very important. I appreciate our speakers staying online. And then I would just, again, let's continue the conversation. If you've thought of anything, and also I would say to the two of you or all four of you, if you have questions among yourself that you want to bring back to us, that would be great also. Questions? Sure. Go ahead, Sarah.
I had a small question, not maybe a really broad scope, but maybe I think it's directed at, for starters, at least to Dr. Rice. You mentioned a lot about, the context of soil microbial diversity and loss of diversity. And one of your questions in that synthesis of like, what are the goals and what questions we need to be asking was, do we know the loss of the scope of the genetic resource loss? How would you, when you asked that question, I was thinking of it in contrast to how much we do know about topsoil loss and carbon loss and about biodiversity loss for macroorganisms globally. How would you characterize our state of knowledge about soil biodiversity loss?
Oh, there. Yeah, okay. I'll push that back. I should know this. I've been here for so many years. We don't know. I don't think we know that much about the loss and potential recovery. I just had a PhD defense, not my student, but I was on the committee, and that was one of the questions. He made the statement that in the system in native prairie, that over long-term 30, 40 years of certain treatment, he said there was a loss of taxa, but then it recovered in 3 years, or a new taxa. So my question was, did we lose it? Was it always there but non-detectable? And then under the right conditions, it came back in higher abundance that be detectable. So that probably, you can figure out how I interpreted it, but so we don't know. And he cited a few studies, but I don't think we know. Is it just because we can't detect that gene abundance, and then the conditions become right? That's my presumption. Kate or some others can disagree with me, but I question whether our detection levels and then we provide the right soil environmental conditions for that. We know that when you change to no-till systems that we get functionally, okay, phospholipids and other enzyme characteristics that we can recover in our data, it shows 3 or 5 years. So were those organisms still there, or they started producing those products, or were they recruited back into the system? So I guess I don't think we know. That was my question.
Yes, Elva.
When talking to farmers, they seem to all stress both inoculations,
Yeah.
and there's a lot of products out there. But another thing they often bring up is that they want to foster fungi. So they want to promote fungi to bacteria ratio. I've worked in soils and we've doubled soil organic matter. Haven't seen, and completely changed bacterial and fungal communities. Haven't seen a change in fungal and bacterial ratios. So I'm kind of curious what your view on that is. A more healthy soil ecosystem one that is fungal dominated?
Yeah, so you're right. We don't see a huge change in ratios, but we do see certain groups change quite a bit. And since I compare grasslands and ag systems, I use grasslands as my model. It's being climate resilient, nutrient efficient, and water efficient. Can we make ag systems look more like the prairie, or act more like the prairie? And our work would suggest that yes, the fungi are really key. We see an increase in the fungi with less disturbed systems. They're not exactly like the native prairie, but we get more fungi, we get more arbuscular mycorrhizal fungi, and then we get the associated aggregation in that. My analogy is that in tilled systems, it's like the microbes, the bacteria in particular, it's like going into a candy store. They get that tillage, they get a sugar high, and they crash and burn. And in a no-till system or less disturbed system, that there's more evenness in the carbon substrate availability. And so that tends to favor actinomycetes and the fungi. But we do see some changes in the ratios. There's a tendency to increase, but not a lot of times it's not significant. But then if we look at particularly the arbuscular mycorrhizal fungi, they show up.
Other questions? Use your speaker, please.
Your mic.
You said you had 3 favorite indicators of activity in your trifecta.
Yeah.
What were they?
So actually, my favorite is phospholipids because
Biomass.
it determines the functional groups, the gram-negative, gram-positive. It picks up the arbuscular mycorrhizae, and that seems to be, and fungi. And going back to that previous question, that picks up that early indicator. We can see changes within 3-5 years, certainly in our research trials, but even on our on-farm network setup. And that seems to be fairly consistent. And then I can graph fungi or AMF to aggregate, and it's pretty linear. So I like phospholipids. It's not easy to do, but not too bad.
Yeah.
And there's commercial labs
you
doing
can
it.
send it out.
Right. And not cheap. And then I was not a big fan of enzymes, but I'm becoming a better fan of enzymes. Beta glucosidase. NAG doesn't, I know Wayne talked about NAG. He can say it better than I can. But the nitrogen cycling enzyme is okay, but it's influenced by fertilizer. And same thing with phosphatase. And so it seems beta glucosidase seems to be our more consistent indicator, at least the way we think things are going. And then I guess it's those two.
Okay.
I don't think the genomics is prime time yet. It's expensive. And again, there's genes there, but I'm a functional guy, right? I want to know what they're doing and how they're living. And that just tells you potential. And I don't know if we're ready to interpret that data yet. In scientific, yes, but to give a farmer, farmers have given me data and I'm going, yeah, you got this gene, I can't, I don't think we have enough scientific basis to make any interpretation behind that. So that's.
Further questions? Yes, Chuck.
I'd like to ask the question about the vegetables and washing or lack of washing of products, fruit and vegetables. I guess when I was on the board, we toured Salinas Valley in Arizona, and I guess in a more intensive agriculture system or more confined system where we got a lot more people than the hunter and gatherers, with organic material being applied to those systems, we've had outbreaks of salmonella and E. coli and all that. And I guess my question is, in a less extensive society, the lack of washing might be okay, but I worry about in a more intensive system, as far as looking at that transfer of those maybe not native soil microbes to the gut. Any thoughts on that?
Yeah, so everything, and thank you for that question. Everything has, pros and cons, and of course that risk is there, and that is probably one reason why hunter-gatherers wouldn't live as long as we do, because our hygiene and the way we live today is different. So, but I was really coming to it from the perspective of, our gut microbiome, the total alpha diversity has gone down significantly. And with healthy diet and lifestyle practices, we can probably prevent further reduction, but that is definitely not going to improve the alpha diversity. And one option is to, if we can, and again, there is so little data, and obviously when more research will be done, this will be a significant barrier that researchers would need to address, is how do we get the good ones and eliminate the bad ones, which would be pathogenic. So again, that's something that requires a lot of research and investigation. And I don't think the scientific literature has enough information based on which we know how exactly to do it. But and we don't even know that this can be done in a large scale in a modern society, Western society. We don't know that. But if we don't have that, if we cannot accomplish that, then what is another way to improve our alpha diversity? It's more a question than really a solution here. So.
I have, no, I'm sorry.
Sorry, I'll answer that just from a slightly different angle. So I think if you are trying to increase biodiversity of your gut by eating fruits and vegetables, you think about what washing does is it doesn't remove the majority of organisms on the product anyway. And when you have an outbreak, it's because it's a numbers game and you've had some incidence that has occurred so that you have more of the pathogen than you want. And for things like pathogenic E. coli, that's maybe more than one cell, right? So one cell can cause illness. Same for some of those different types of salmonella. So I think if you want to encourage the diversity, I think you can do that by just encouraging eating fruits and vegetables, because you don't wash everything off, and the ones you want for that diversity, I think you will continue to increase.
Wayne, you had a comment.
Yeah, sorry, I was going to change topics. Is that all right to change topics?
Sure.
Thank you. I guess I was just kind of intrigued by one of your earlier conversations at the beginning of the meeting when you were framing, kind of the charge for the committee and around the discussion of hydroponics. And I know that is kind of I would say kind of a politically charged, and I'm sure you're well aware of the politically charged nature of that too. And in terms of organic certification and those things. And so, and I'm not trying to persuade the committee one way or the other. It's up to you all, you know, what your focus is. I would just say that from my perspective, that it's interesting, you know, early on, I think that the concept of human health was more of just looking at kind of physical well-being as kind of defined by the World Health Organization. But now, since I think it's like, I'm just looking up 2015, they basically changed their definition to be more holistic and more cognizant of the ecosystems and the relationship to the ecology, and as an underpinning kind of component of health and those types of drivers. And so they're kind of thinking more holistically about it. And the reason I say that is because as challenging as we are finding it to draw the connections between soil health and human health through the nutrition pathway, I feel like it's really right in front of us and already well established, that connection between the practices that improve soil health and the environmental benefits that come with it. And those healthy, you know, human health components of those environmental benefits. Because we know that, you know, when we're looking at the soil health, that a lot of those practices show many times, you know, at many scales, from plot scales to watershed scales, to reduce nutrient loss to the waterways, reduce, you know, leaching to groundwater. And of course, we're depending on a lot of those practices to help us both mitigate and adapt to climate change too, increasing carbon sequestration, all those things. So I would just ask that you consider that when you are debating whether to expand this to those types of systems.
Thank you very much. We have a committee member, Mike.
Thanks. I just following up on Wayne's reintroduction of the hydroponics issue. I just, you know, want to make sure we keep in mind that, you know, hydroponic systems, whether they're used, you know, commercially, they're not sterile. I mean, there's still microbes associated with the roots, with the plants involved. So, you know, I know we're talking about soil in a more you know, broad perspective here, but that whole issue of these other ways of growing crops, you know, they're not grown under necessarily sterile conditions. And so, I mean, somewhat in some cases, they may be trying to do that, but there's still always microbes there in some way. And so there's still that transmission possibility into the foods and so forth.
Thank you. Any further comments? I would just like to throw in my little bias here, which is that in soils, there is a tremendous food web that feeds on microbes, and they're all the invertebrates, and the invertebrates and how they interact contribute also to soil structure, to placement of soil carbon. And I think that I'll come back in just a second. Let me get my little sentence said. And I just think that one of the things that we have to consider is whether in the future, not within the statement of task of this committee perhaps, but are we looking at soil holistically? We say we want to look at it from a system perspective, but these invertebrates are also turning over carbon and nitrogen and helping to store carbon in soil, as well as contributing to ecosystem services. And so I think in the future, we may be thinking about soil health as a holistic soil health. And so now.
Oh, you're on mute.
And sorry, I turned it off.
I've been thinking about this the whole time, and I think there was enough points made that I'm going to throw it out there for people. And Chuck knows we've spent a lot of time here trying to define the term wellness and what it means in humans and how to measure it. And it's sort of evolved to be sort of this resilience measure, but now we're talking about soil wellness here as well, and how that's different from health. So I think where we've gotten to with health is it's sort of something you can correct, and it's the absence of any disease or defined deficiency or something. I'm wondering if some of these discussions are delineating the difference between just soil health and soil wellness, which would reflect all this holistic aspect and the resilience of the soil over time, its resistance to all these perturbations that it's hit with, because that's, you know, from the human, we think about it as if you have an immune challenge, a well person is able to resolve it, has those resources available to them. I'm wondering if some of this discussion is kind of at the soil wellness, human wellness interface, the way some of these terms have come in. So I just going to throw that out there if we want to talk about health versus wellness.
Thanks very much. I think that's a great point for us to consider as what we're defining. Did you have a comment on that, Chuck?
Just to follow up, if it's okay, I mean, part of the reason we have pathogens is that they're opportunistic, right? And there's something there. They're always there, but they're biologically controlled. And so, you know, the whole aspect of biocontrol is you got a competitive ecosystem, and the healthy microbes are there, but then there's an opportunity for a pathogen to come in and take advantage of that niche. And so I think having whatever healthy soil is keeping those pathogens in control. They're there for the most part, but then it just they're taking advantage of a certain situation.
Thank you. I think if there are no further questions, we're going to stop and thank our speakers once again. And to all of you online and watching it live, I want to thank you very much. The committee, as we think about this statement of task and what we're going to be doing as a committee, it's really important. The committee will hold its next public meeting on May 16th. And as mentioned before, as more information about the agenda comes available, we will be putting that on the website, so you'll be able to find that. And I want to thank everybody here and online for attending today's session. And I really thank the speakers very much, both online and present.